So that was about the vegetative propagation by stem. Now we move to the next one, that is reproduction by leaves. So that will be the modified leaves which gives rise to the new plant. So leaves of some plants such as Brophyllum produce buds in the notches in their margins. So the leaf are such way. Suppose if this is a leaf over here, now this leaf will be having some kind of a bud. So these are the buds which are going to be there in their margins. Margins is the edge of the leaves are called as the margins. So there will be like this bud-like structure over there, which is called, which is over here as in case of Bryophyllum. So Bryophyllum is a kind of a plant which has got such kind of leaves. Now when such leaves fall, okay, so what is going to happen? The leaves are going to be permanently on that particular plant. So when the leaf is going to become dry and it is going to fall, at that time when leaf falls in moist soil, their buds in the margin begin to grow as a tiny plant. So this is just going to be that this when they are fall, they are not going to see. In case of spore formation, we learned that the buds were there, but their spores were just carried away by the wind, and hence they were like seed dispersal taking place. Here it is not going to be seed dispersal, it is not going to fall down by itself. It will be when the leaf is going to fall down, in the fall season, or whenever the leaf becomes dry, it is going to fall down. When it falls down, and if it falls down in a moist kind of area, then it is going to have the favorable kind of conditions and it is going to grow into a new plant. So that's why we have got the modified leaves as the kind of vegetative propagation. So vegetative propagation by leaves is in case of Bryophyllum. So Bryophyllum is an example over here. So you have got over here, take a full grown leaf of Bryophyllum with some adventitious roots but growing out into the margin. Cut the leaf into bits, each containing growing adventitious bud. So you can cut it even a small piece like this much over here. You can cut it and place it in the. So put these bits in a flower pot and cover them under soil, moist soil, keeping the emerging bud just protecting out from the surface. Observe after 10 to 12 days, tiny plants grow out from the buds. So conclusion: Bryophyllum is a plant whose leaves produce adventitious buds in their margins. So adventitious are the kind of roots, that's why it's called adventitious. So it is going to be like this and the roots are going to be like this, okay. There are two kinds of roots actually, if you know it. One is the root which will be like this. So if the root is something like this, it is something like this, then adventitious. And if the root is like this, then it is called as taproot. So taproot and adventitious roots. In this case, it is adventitious roots. So adventitious buds grow into the new plant under favorable conditions when the leaf with buds fall off from the parent plant. So that was about the Bryophyllum. Now we come to the next part, and that is called as reproduction by roots. So the third one is the reproduction by roots. So the last one for the vegetative propagation is the roots. Plants like sweet potato and asparagus develop numerous large swollen fleshy roots. These roots contain a large quantity of food inside them. Each such single root is capable of giving rise to a new plant. Carrot, a taproot, is a biennial plant. Its roots grow vegetatively in the first year and store food. In the following year, its stem produces flowers and seeds which die by the end of the year. Buds produced at the base of the old stem just above the taproot are meant for vegetative propagation. So you can see over here that in case of reproduction plants of roots, the first thing is about the sweet potato or asparagus, wherein they are not only, the roots are not only going to be the one which is going to store the food for it, but is also going to be a vegetative part. So vegetative part, so such a thing are capable of giving rise to a new plant. In case of a carrot, now carrot is a biennial plant. What do you mean by biennial plant? A plant which has the life of only 2 years. Okay, so that means it is going to be there it is going to be existing only for 2 years, and then a new plant will be required for the growth of the carrot. So that's why the carrot plant, if you grow it in your garden, it will, or in a pot, you will find that it will be there only for 2 years, and then it will die off by itself. So in this case, it is seen that in the first year it grows vegetatively in the first year and stores food. So the first thing, in first year it is going to grow many carrots and it is going to store the food over there, and in the following year the stem produces flowers and seeds which die by the end of the year, and the buds produce at the base. So at the base there will be, of the old stem, there will be buds produced which will grow into new plants. So that will be the tap roots meant for vegetative propagation. So that is about the carrot. And you have seen over here, we had seen about the grass also, about the grass being the stem being the vegetative part. So here they are given the activity 5, which is about the grass. That is, take a piece of lawn grass in a pot and cover some of it portion with soil. Leave it for 10-12 days. Sprinkle some water every day over the soil so as to keep it moist. After a few days, new grass shoots grow out from the buds of the nodes. So common lawn grass has long internodes between successive nodes creeping onto the surface of the ground. From each node, roots are given out. The axillary bud at the nodes produce new shoots. So this is just about the grass, which is not connected to the roots, but it is connected to the stem, which we had learned earlier. So in this way, the vegetative propagation is going to take place from either the stem, leaves, or the roots. So when we talk about, or when we try to see the advantages, so there are the vegetative kind of propagation is very advantageous in certain manner, and of course there are some disadvantages also. So if we see the advantages, let's see what are the advantages. Farmers prefer to grow certain crops by vegetative method. The advantages in doing so is as follows. Reproduction by vegetative plants takes place in a shorter time. See, reproduction is very short. फटाफट से हो जाता है. Okay, it's a very fast process. Why? Because नेतर क्या है, सीड को सो किया, सीड तो पहले वायबल है कि नहीं है, उसके बाद उसको सो किया, फिर वो ग्रो होगा, पहले छोटा प्लांट फिर बड़ा प्लांट होगा, तो it takes a long time. Whereas the vegetative parts का जो भी रहेगा, it is quite fast. So that's why it is going to be reproduction takes place in shorter time. New plants thus spread very fast in a small area because the new plant क्या होगा, इसका root is going to give rise to another plant, that is going to give rise to another plant, that another plant, and so on. So you can see that vegetative propagation वाले जितने भी plants are, they are going to grow much faster and going to cover a particular area much faster and spread faster in a small area. Thirdly, it is a surer method. You don't need to see, अरे ये seed अच्छा है कि नहीं है, क्या होगा पता नहीं है, whether it is going to grow or it not going to grow. So for 3-4 days you are just seeing that, अरे it is whether the sprouts are coming out or not. So such things are not required. You are sure that if you take this bud, if you take this particular node, internode, if you take this particular leaf or stem, it is going to grow into a new plant for sure. That's why it is a surer method. Fourth and very important is that all the good characters of the mother plant are retained by the daughter plant. See, it is a part of the original plant. Okay, it is a part of the original plant, so that's why whatever is going to grow out of this particular part of the plant will be exactly identical to what was the original mother plant. Okay, if you take a carrot of a particular, if you take a particular carrot, then the plant which will be growing will be also of the same kind that the carrot of which you have taken. If the carrot was long and reddish, so automatically the one which will be grown will be red and long. Okay, if it is like orange and thicker one, then it will be of that kind. So you know that what kind of thing is going to be generated by this thing. So hence we see the good characters of the mothers are retained by the daughter plant. Of course, there are disadvantages. As all plants developed by vegetative reproduction are identical, they are likely to be affected simultaneously if a disease spreads in the farm. See, it is like having all the eggs in the same basket. Okay, if all of them in the same basket, अगर एक को कुछ हुआ तो बाकी सबको भी साथ में होने ही वाला है. It is that particular kind of a proverb. So Similarly over here, if there is a complete farm which has got the same kind of vegetation throughout and all are just being vegetative, so all of them of the same family in fact, then if one of the part gets diseased, automatically it is going to spread throughout in the whole area. So that's why the spreading of the disease will be much faster in case of such a kind of a condition, because they are going to spread within the in the farm, simultaneously in the farm. Second is dispersal of plants does not take place on its own. So daughter plants so developed tend to remain nearby and are restricted to a particular area, leading to competition for resources. This is very important. Let's understand this thing properly. What is happening is, a particular area, there are only, let's consider that there are only carrots growing over there. Now each and every plant requires a certain amount of nutrients from the soil. The amount of nutrients present in the soil is limited. Okay. Now each and every plant, if every plant is requiring only one kind of nutrient from the soil, then automatically there will be a scarcity of that particular resource in that soil. But if there would have been different kinds of plants over there, then each plant would require different kind of nutrient, and hence the nutrient required for the plant will not be so difficult. It would not be difficult for the plant to take these resources from the soil. Hence you can see that when the plants are of the same kind in that particular vicinity, in that particular area, then there will be a competition to extract the nutrients from the soil. So the resources of the nutrients will fall short, and hence there will be a competition for within that particular plants to extract the nutrients, and they may get deteriorated in their quality. अगर समझ में नहीं है तो I'll give you a very simple example. Suppose I got 3 bowls of chocolates over here. One was gems, okay, another was chewing gums, and the third one maybe you can say some kind of Cadbury. So that was just pieces of Cadbury in the bowls. Now if there are 10 students in the class and each and every one is going to be told that okay, take whatever you want from here, but it would be that one person will be taking only one of them. Now suppose everybody wants to take only gems, then what will happen? That bowl of gems will be too insufficient for all the 10 students. Or even if they get, each and every get something, the amount of gems or they will be getting will be quite less, because suppose there are 100 gems over there, so 10 students will get 10 each. But suppose it is this way, that out of the 10, 3 said nahi humko to chewing gum chahiye, and 3 says that we want that Cadbury, then what will happen? 100 of this, 100 of this, and 100 of this will be divided in such a way that everybody, means hardly 3 people are going to share one bowl. That means each will get at least 30 or 35 each. So that's the reason you can see that the nutrients are available to all the students in a large quantity if they are requiring different nutrients. So that is exactly what happens when the plants of the same kind are grown throughout that particular soil. So that's the thing what they are given here. Dispersal of plants does not take place on its own. See, the plant is not going to be the, अभी क्या होगा, जो भी plant है उसका vegetative part उधर ही गिरेगा, उधर ही वो नया plant बनेगा. ऐसा तो है नहीं that it is going to fly off and go to 1 kilometer away and then it is going to get a new plant. No. So it is going to be within that particular vicinity only. So daughter plants so developed tend to remain nearby. They are just going to be nearby the mother plant and restrict a particular area. They will be restricted to a particular area. I cannot go within more than a particular area, leading to competition for resources. So resources will be short of in that particular thing, because I told you everybody was going for gems, whereas the other 2 nutrients are still remaining over there because nobody wanted to opt for it. So here also the same thing. The nutrients, each and every plant requires the same kind of nutrients, so it will be difficult for them to get those, and hence there will be scarcity of the resources. So that was how we had the vegetative propagation or the asexual method of reproduction naturally. Okay, this was the vegetative reproduction naturally. Now we are going to see the next kind of vegetative reproduction, but that is artificial vegetative reproduction, wherein we are going to use some method for reproduction of that particular plant. Okay. So we see over here, the next one is artificial vegetative propagation. So it is artificial vegetative propagation. Okay. Now, so nowadays farmers, the horticulturists, they develop many kinds of artificial method by which a plant can be grown into a new plant. So that's why these days farmers and horticulturists have developed certain artificial methods of vegetative propagation, such as cutting, layering, grafting and tissue culture. So these are the different kinds of the artificial vegetative propagation. The first one being cutting. It's a very simple method. Suppose I come to your place, and at your place I can see a very beautiful plant of rose. Okay. So I just say, oh wow, what a beautiful plant of rose. मेरे घर में तो इतना अच्छा rose grow ही नहीं होता. So what you do is just you take one of the stem of that plant, cut it and just give it to me. Maybe it may have the rose or it may not have the rose, doesn't matter at all. It's just a stem of that particular part. And then I take that stem, come to my house, put it in water. Within few days you will find that the lower part which was kept in water will develop into roots. And once the roots are developed, you can place the plant into the soil and grow into a new plant, which will be exactly identical to the plant which was there in your place, from where I cut, you gave me that particular stem. So this is how you can do cutting. So cutting is a very simple method. In this method, the stem is cut into small pieces with each bearing an axillary bud. So the only requirement is that whatever is part of the stem we are cutting should have an axillary bud. So it should have that particular bud like thing from where the leaves can be grown. The cut ends are planted in moist soil. After a few days, they strike roots and grow into new plants. This method is normally employed for propagating plants like sugarcane, rose, China rose, lemon, etc. Now in this particular chapter you will have to learn the examples because they are going to be very, very important. You need to know that what are the examples of what kind of vegetative propagation. So you need to learn the method as well as you need to learn the different examples of that particular method. So first one in the vegetative propagation was cutting, a very, very easy thing, that you are just going to cut a part of the stem which is having an auxiliary bud. Auxiliary bud के साथ में उसको soil में डाला, it will grow into a root and grow into a new plant. The next one is layering. Now layering is a kind of a thing in which, suppose this is a plant over here, so this is a plant, so this is one over here, this is another stem over here, this is plant like this. So what you do is that, suppose this is a stem over here, this stem, you are just going to bend it, so this is the ground level, you are just going to bend it such that this is going to bend like this and come out like this. So these are the leaves over here and these are the leaves. So here, before you put this into the ground, you are just going to remove the brown colored scaling on top of it, and you will find that few days later there will be roots which will be grown over here. You cut it from here and this becomes a individual plant by itself. So this is how you can have a layering done. So in this method, a portion of one of the lower branch of the plant with auxiliary bud and at node is bent down into the ground so that it touches the soil. Okay, so this was bent down so that it touches the soil. Okay. A ring or bark is removed from its portion which is then covered with soil. So here what we are doing that वो brown color का जो ऊपर का जो part रहता है उसको हम लोग छील देंगे. We'll just going to remove that thing. You can remove that thing so that you can have the white part inside. So that there is a, see the upper part, white part is going to be hard and it is a protective layer. So that protective layer will not allow the roots to grow because they will be protecting the plant from the soil and its content. So once you remove the protective layer, then it is able to take the nutrients from the soil and start the roots development over there. So some heavy object such as a piece of stone or a brick is kept on the branch so that it does not come out of the soil. So you are going to put it over here, then you are going to place something so that upar ut nahi jaye. Otherwise it will have tendency to come back to its original position. In a few days, when the branch gives out roots, it is cut off from the main plant. It then continues to grow out as an independent plant. This method is used for propagation of plants like mint, rose, jasmine, etc. So here we can see that you have first what we did was वो plant था, plant का ये stem ऐसे था, okay? You are bending it over here. The branch is bent over here. This part over here is cleared off. It has gone into the soil. Here you are just उसको ऊपर से छील देते हैं, okay? So that the outer covering is removed, a ring is cut, and then you are going to place it over here. A new root is going to be developed, and once the new roots are developed, you can cut it from here and this becomes a separate plant altogether. So this is normally done for mint, rose and jasmine. So the jasmine, etc. These are the examples for layering. So the next thing over here was layering. Okay, very easy. The third one is grafting. Now in case of grafting, it's a very unique thing because here what we are doing is that we are clubbing two different plants. Sometimes we can do even with different plants that the root system is of a different plant and the shoot is of a different plant. Okay, so suppose there is some particular plant which is not having a proper shoot system but the root system is proper, you can cut it down. So you will be having the root system over here like this. So this is the root system over here. You cut this particular part over here and make a V cut inside, and then you take another plant, okay, किसी और plant tree में से कहीं से भी और एक branch निकाला, and that branch you cut it this way. So this will be having a cut section like this. So these are the already existing leaves over here. You place this inside over here like this. So this plant will be like this over here, like this, and this will be the leaves over here. And then you are going to tape it up. You are going to tape it up and put some wax over here so that it doesn't dry off. See, because once you cut this thing, it is not having any water inside. So water जितना है उतना उसको retain करने के लिए till the time the new, so it is like a fracture. So fracture जब हम लोग replace करते हैं उसके बाद में ही सब conduction वगैरह हो पाएगा. So जब तक वो fracture repair नहीं होता है तब तक ये dry नहीं हो, इसलिए we are putting wax over here so that it is not evaporating, it is not getting dry. And you will find that this is going to connect with each other. So this particular, if I take this particular cut over here, so this is going to connect with each other automatically, and the water which is coming from the roots is going to enter into this thing, and the plant is going to survive properly and form into a new plant altogether. So this is how the grafting is done. In the grafting, the upper part over here, this is called as the scion, and this is called as the stalk. So they are the two parts. The root system is the stalk and the shoot system is the scion. So we see over here grafting. In some cases, such as rose, mango, guava, etc., a small shoot or bud of a desired variety of a plant is intimately fixed to the stem of another of the same or related species. Okay, so same or related species का एक प्लांट में से ही कुछ निकाल के वहां पे बड उसका उधर intimately fix किया उसको, very tightly fixed with the original plant. The plant receiving the bud or the shoot is called as the stalk. This is called as the stalk, sorry, the stalk, not the stalk. Is the stalk, and the shoot fixed on it is the scion. For a successful grafting, it is important that the cambium layer of the stalk and the a scan must come into very close contact so that growth may continue. So this is the cambium layer, this is the inner part, so that is the cambium layer, the inner part is the cambium layer. So it should be this way that ये अंदर पूरा बराबर से घुस जाए. Okay, so this is a cut form, and this is the, so this is the stalk and this is the scan. So the scan goes over here, inside over here, so that there is a complete gap. This gap filling is proper, and it is not able to it is able to have a flow of the nutrients from the lower part to the upper part. The grafted point are then bound together with a tape, and the joint is covered with a wax to prevent dehydration and is protected from bacterial infection. So this particular part is taped, it is put some wax over here so that it is sealed, so that it is not getting dried out, dehydration nahi ho, and it is also protecting from bacterial infections. In a few days, the new cells develop and the new plant grows. Okay, so the new plant starts growing. You will be surprised to know that in one of the, and I don't know exactly the place where it is located, but one person has done so much grafting on a mango tree that one original mango tree has now got 125 different graftings on the same tree, that tree is producing 125 different varieties of mango on the same tree. Okay, somewhere in Gujarat was there. I read it long back, so I don't remember what is exactly the name of the place. But there is one particular person who did 125 different kinds of grafting on the same mango tree, that each and every plant which is going, each and every branch of that tree would be different kind of mangoes growing on it. So remarkable, you can see. But yes, of course you cannot have an apple on a mango tree, so that has to be a limitation. It will be of the same species or similar kind. So that was about the grafting. Now next one is micropropagation. So micropropagation. Now this micropropagation is actually the one in which is normally done in laboratories. So micropropagation. Now in this case, a small kind of a bud or the shoot part was taken in, the apex ka part is taken in, very small part, and it is put in a beaker or a flask containing some nutrients. So when it is kept over there in favorable conditions which are given to it artificially in a laboratory, it will grow into a mass of plant which is going to form into a plantlet, and then that can be sown into the soil to form the complete plant. So this kind of a thing is called as micropropagation, which is normally done in case of science labs. So if it is the propagation of plant by cell and tissue culture. So this kind of a thing is called as the cell and tissue culture. If vegetative propagation is not possible in a crop, then buds, shoot, apex or any other part of the plant can be used as explant for micropropagation. The explants are treated with sterilized chemicals to prevent microbial growth, and they cultured in a particular nutrient medium. So as I told you, the bud or the apex, apical bud or any kind of a vegetative part is going to be taken over there. It is going to be put into a antibacterial fat so that the bacterial growth is not taking place, and put into the nutrients wherein it can grow into a plantlet. So the first one, the small piece which is cut out is called an explant. Okay, the small piece cut out as explant. So cells grow and divide into form a cell mass called as the callus. So the explant, after some few days, is going to become a callus. The callus is the one which is the mass of cells. Some growth regulators, plant hormones are added. The callus differentiates into plant parts looking like a tiny plant called as a plantlet. So within a short duration of time, the callus will start growing into some small roots and some small leaf-like structure, and that particular plant is then called as a plantlet. So after 4 to 6 weeks, the plantlets are transferred into the soil. This can be used to derive and multiply disease-free stalks. This is kind of tissue culture technique. It is being used in orchids, gladiolus, chrysanthemum, potato, sugarcane, etc. Okay, these are the different names which are of the different kind of high yielding plants. Benefits of tissue culture: it provides rapid propagation of identical individuals. This technique is very productive for superior variety. So when you want a very superior variety of certain things, people will go, the agriculturists will go for a micropropagation method. It can be applied to interspecific hybrids. So if there is some hybrid, now hybrids are something like, you must have seen that initially the rose were only of one color, red color. Now it is of different colors. The tulips are of different colors. So what are those? They are basically hybrids. So that is the cross pollination between two different kinds of roses. So of course roses don't do cross pollination, but if they are trying to make something like hybrids in a kind of a laboratory. So interspecific hybrids are going to be formed. It is very useful in cases where seeds are dormant. In these, the embryo can be cultured and micropropagated. So wherever the seeds, see, many times the seeds are, even if there are many seeds available, but they will not grow till a certain number of years. So they are called as dormancy. That particular period in which till the time the seed is not going to mature enough to form into a new plant is called as dormancy. So if the seeds are dormant and they want to propagate them, they can use this method by using the micropropagation. But there are limitations, of course. The limitation will be, it cannot be applied to all cases. Of course, it cannot be applied to all the cases. It will be only a certain cases where it can be applied. And it is not easily applicable in remote agricultural areas. In remote agricultural areas, they do not have these facilities that उनके पास lab हो जहाँ पे वो सब micropropagation कर सके. So that particular thing is a disadvantage of or limitations of the tissue culture or the micropropagation. So in this case, we see that the vegetative reproduction can be taken artificially by 4 methods: cutting, that is just cutting the stem and placing it over there; layering, that is the one in which you are bending the thing into the soil and the roots starts growing over there; grafting, which is this method, the scan and the stalk fixed on top of each other and taped it up; and micropropagation is by the taking a small bud or the axial part of the plant. Okay, so that is the different types of reproduction, asexual reproduction. So we completed the asexual reproduction of plants by this. Now in the next video, that is next week, we will be taking up the sexual reproduction in plants, that is study of the flowers and the seeds.